Hi everybody, it's Daniel here, a fellow student of yours from CMCC. I wanted to take a little bit of your time today to talk about a topic of our course biochemistry that I find very important yet somewhat difficult to understand at times, and that is cellular respiration. It contains glycolysis, the citric acid cycle, and the electron transport chain, and essentially it's the concept of taking the food we eat and turning it into energy. This will be, I believe, a central focus of our course, and it will be really helpful to understand these processes. Before we begin, I just want you all to understand that this is only made as an accessory to our studies, to the lectures that we attend, and not a supplement. So please still use your lecture material, please focus on that, and only use this as a study aid. Alright, so let's just jump into this. When we look at a diagram like this, it makes us want to jump out of our seat and leave. However, at the end of this, I want us to have the understanding of this in our minds to look like something like this. It contains all of the intricate components of cellular respiration from this very scary concept map in a very simple yet concise yet complicated diagram here that is just neat and tidy. Like I said before, cellular respiration is the process of turning our food into energy. But let's focus on glucose because carbohydrates, they provide us with glucose. Like I mentioned before, cellular respiration, it's composed of glycolysis, the citric acid cycle, and I'm going to just write Ca for that, and the electron transport chain, which I'm also going to write ETC. We'll cover these in later topics. Okay. All of these are commonly denoted through the following equation. It's a chemical structure equation, and it's not meant to scare anybody or confuse anybody, but it really just puts everything in a nutshell. And I want to start from the basics, and we'll build from there. This is just an introduction to glycolysis. So, cellular respiration or metabolism of energy looks a little bit like this. We have glucose, I'm just going to say GLU for now, plus oxygen, giving us water, giving us carbon dioxide, and giving us energy. Okay? Now, just to be specific, I'm going to write this down as a chemical structure. So we have C6H12O6, that's glucose, C6H12O6, plus 6O2, okay, plus 6H2O, plus 6CO2, plus ATP. Now, sometimes, or most of the time, we say we have 36 ATP. Other times, it's argued that there's 38 ATP produced, and that really changes from text to text depending on certain semantics and other things. For now, let's just talk about ATP as energy produced from this. Now that we know this, let's move on and talk about the significance of ATP and why it produces energy. ATP gives us energy because of its chemical structure. So here is a diagram of ATP. When we look at ATP, we see that there are subgroups within it, functional groups or sections. So to make this a little bit more easy for us to look at, I have a colored version that's labeled. We have 3 subgroups. One of the subgroups has 3 sections to it, but let's go through this slowly. The first group we have is adenine. that's the beginning of the name of ATP, adenosine triphosphate. Adenine is attached to a sugar that is a ribose sugar, and we learned about those. That's actually what gives it the name adenosine. Now, as you would expect, there are 3 phosphate groups here, which gives us the name triphosphate. Labeled in green, yellow, and purple, we're seeing that if there were only one phosphate, it would be adenosine monophosphate, or AMP. You will see this. If only one group was lost, we would have ADP, which is adenosine diphosphate. And when we have all 3, we have good old ATP. Now, when we look at ATP, we have to understand that its chemical structure is what provides the energy. And how? You have to know what it is about ATP. A lot of people just think that there's something special about ATP, but we don't really understand, or we need to be a little bit more specific with what it is about ATP that gives so much energy. And I'm going to discuss that right now. What gives ATP all of its energy, or the most of its energy, are their high energy bonds right here. That is between the phosphate group beside the adjacent phosphate group. The highest energy bond would be between the second and third phosphate group. This has a lot of energy. It wants to get off of ATP, this third phosphate group, and when this happens, energy is released. We also have to understand that molecules like to be stable. So whenever there is an opportunity to do so, molecules will take it. Let us take a closer look at phosphate. A phosphate group here, as you can see, is connected with a bond to the oxygen group of the phosphate beside it. But typically, if there was no oxygen here, the phosphate group would simply have an R group right here. That just symbolizes any other extension. It can be any other molecule as long as it's chemically compatible. When we look at glycolysis, we'll have a better understanding, step by step, how phosphate plays a very huge role in the 10 steps of glycolysis. But for now, what I want you to understand is that within this bond here, a lot of energy is released when we break it. This is a general schematic of glycolysis. Now, I'm only going to cover a few things generally and the first stage of glycolysis as well. After that, we'll take a break until the next time. First, let's just talk about why glycolysis occurs the way it does. We have this molecule, which is glucose. It was digested from a carbohydrate, and it was brought into our blood, and it was processed, and it was brought forth to our cells for cellular respiration. It's a very high energy molecule, but it's extremely complicated, and you can see that here. It's a molecule with 6 carbons, and throughout this process, I want you to count the carbons. We have carbon 1, 2, 3, 4, 5, and 6. You will see throughout glycolysis these carbons decreasing and changing. And what we want to understand is that we are trying to find a way to optimize the amount of energy released. In other words, we want to find the most efficient way to make the most out of glucose, because our bodies are not certain when and when we won't have available energy, so it wants to optimize that for the maximum efficiency. Now, I want you to imagine glucose as this energy molecule. It's a precursor for energy. It gives us a lot of ATP, but we have to understand that you cannot do this in just one step. It's like having a vat of gasoline. What would last longer? Lighting that entire vat of gasoline at once, or using a drip onto a table and lighting that? My guess is the drip would last a lot longer, and we would make a longer burning time from that gasoline as opposed to just burning it all at once. That is why when we describe glycolysis, we describe it as something called a stepwise catabolic reaction. Stepwise, so you can imagine steps, and catabolic reaction. Catabolic means to break. It means to break something that is large into many small components. This is a catabolic reaction. The opposite would be an anabolic reaction, which takes many small components and turns it into one large one. Keeping this in mind, now we have a better idea as to why glycolysis has so many steps. If we were to use the energy of glycolysis, of glucose, in one step, we would waste a lot of energy to heat, to light maybe, and just to the rest of the universe. And that's how in chemistry they describe energy laws. But let's look at glucose here and the first step. Actually, before we do that, I just want to make clear one concept within glycolysis, and that is the irreversible reactions. For now, I just want you to pay attention to enzymes that end in kinase. Whenever you see an enzyme in glycolysis that ends in kinase, you know that the reaction is one way and not two-way. Keep this in mind as we go through glycolysis. The first step in glycolysis is attaching a phosphate group to glucose. Whenever we go through steps in glycolysis, I want you to pay attention to the names of the molecules after each step. These names are crucial to what has happened before and after. The processes are all done for a reason, and they're done as a way to maximize energy production. In the first step, we see glucose as a six-membered ring, with the first carbon being located right here. This is the sixth carbon. The sixth carbon is not a member of the five-carbon ring. It is outside, and it's very accessible. Let's assume we have ATP just floating around. And by floating around, I want you to keep in mind that glycolysis does not occur in the mitochondria. It is only the citric acid cycle and electron transport chain that occur in the mitochondria. Glycolysis occurs in the cytosol of the cell. We have our ATP. Again, 3 phosphate groups. It is the third phosphate of the ATP that will detach itself. remember the R group that I told you would be on that phosphate group. That itself would attach to the 6th carbon molecule. When this occurs, we will have a new molecule, which would be glucose 6-phosphate. So I want you to see, here we have the new phosphate group, which left ATP and joined on the 6th carbon. When this occurred, that ATP became adenosine diphosphate. So we would change ATP to ADP because we no longer have this phosphate group because it went here. Now, if I'm going a little bit too fast, please let me know, and for the next tutorial I will slow it down. But I want you to understand and appreciate the names. We started with glucose. After the first stage, we got glucose 6-phosphate. Glucose 6-phosphate. Do you see the relationship here with the name and the location of the first phosphate group attached to glucose? Now, there's something else I want you to notice here. This first step has an enzyme called hexokinase. For now, I don't want you to worry about magnesium here. Let's just look at the hexokinase. 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 A kinase leads to an irreversible reaction in glycolysis. So we know that the first stage in glycolysis is irreversible and it cannot be taken back. With this in mind, we know that a kinase needs ATP to function. It doesn't need ADP, it needs ATP because it donates its phosphate group to the 6th carbon of glucose. Before I go any further, actually we're almost done here, I just want to talk about enzymes for a second. We'll use a little bit of an analogy. We live in Canada, and for about 8 months of the year, there's snow on our driveways, on the ground. Now I want you to imagine all of this snow on the ground to be work. Work and an analogy for the reaction. Let's say, in this case, turning glucose to glucose 6-phosphate is the snow on the ground we want to shovel. Okay. That snow on the ground that we want to shovel is the work we need to do. Let's talk about our hands. Our hands are the ATP. Our hands have the energy and the potential to go and turn the snow on the ground, which is glucose, into the shoveled form on the side of the driveways, which is glucose 6-phosphate. But now I want you to think about this logically. How long do you think it would take to use your hands to shovel your driveway? It will take ages and hours and hours. It will take a very long time, but sure enough, the driveway will be shoveled eventually. What would happen if somebody down the street or right across from you walked up to you and gave you a shovel? All of a sudden, you just turned something that would take you many, many, many hours, probably 15 minutes. That is what we call a catalyst or an enzyme. That would be our shovel. It will speed up a reaction. Now, let's take this analogy just a little bit further. When the snow melts, it's gone. We'll never see it again until the next year. Our hands will no longer be carrying snow, so that's out of the equation. But the shovel will still be the same just seconds after that reaction is over. Just seconds after the job. Which is the same for enzymes and catalysts. They are not used in a reaction. When I say not used, I mean they are not used up. At the end of the reaction, hexokinase will still be there. ATP will not. ATP is gone and we have ADP. We lost an ATP. Glucose is gone because now we have glucose 6-phosphate. But hexokinase is ready to do its job again for another glucose group. And if I was just to quickly, I know I'm taking a bit long here, this is just the last second. If I was just to quickly show you just one more time, an enzyme would look like a little blob like that. This would be hexokinase. Now, on the other hand, we'll have ATP fitting into this blob. Okay? Now, in this blob, we'll also have glucose, just here. Okay? The reaction will have one of the phosphates go in here, and that's all sped up by hexokinase. Then, both of these will be gone, and we will be left with hexokinase one more time. And, on the other hand, we'll have glucose 6-phosphate over here. I hope that makes a little bit of sense to you. The concept of the enzyme and the substrate. In this case, the enzyme is hexokinase. The substrate is what? Glucose and ATP. Oops, sorry. ATP. The product is glucose 6-phosphate. I hope this tutorial helped. All I wanted to do was give you a general idea of how to approach glycolysis and the rest of cellular respiration with a good background that would hopefully help you understand it just a little bit more. If you guys really like this, I can keep on making more tutorials. We do have the rest of glycolysis, which is all of this here, step by step. I can take it tutorial by tutorial if you guys want. Just let me know. Thank you for listening, and see you next time.